All right. Hello, and Academy Indigenous Voices series. And today I am happy to be able to introduce you to Matt Anderson, who is an assistant professor of microbiology and microbial infection and immunity at The Ohio State University. Matt got his PhD from Stanford in 2009, and his research interests are in the evolution of genes and genomes in the major human fungal pathogen Candida albicans, and how those genetic changes contribute to phenotypic diversity. And today he's going to tell us about engaging indigenous communities in microbiome research. Matt, it's all yours.
Great. Thank you for the introduction, Larry. So I'll go ahead and turn on my screen. Okay, so hopefully everybody can see this projection of the title slide. Just want to introduce myself really quickly. Matt Anderson Damado, Tita Lage, Duna de lo Quasti, Ohio State Kalaladi. So I'm over here at Ohio State, Eastern Cherokee, and Ohio State kind of hits the intersect of a lot of different people's land. So just need to acknowledge the fact that this is Miami, Wyandotte, some of the 5 tribes, Seneca land, and this last one is eluding me. Eastern Shawnee. So with that, I'd like to walk through what I'm going to be talking to you today about, which is this idea of approaching indigenous communities on their own terms, specifically around microbiome research, although as we're going to see, these topics are going to be pretty applicable across different kind of research frameworks, but we will delve into some of the microbiome components very specifically. So before we start, I just want to provide you kind of a definition of terms. Because of the international nature, I just wanted to make sure that we're referencing both indigenous as an idea and a framework about global populations that have a connection to place. Their culture and their history is connected to that place despite repeated attempts to be influenced by outside forces or outside entities to change the way that they live day to day and the way that they've established kind of frameworks of organization and governance. And then I'll be using the term Native American or American Indian to describe specifically those indigenous people within the contiguous United States that have established nation-to-nation relationships with the United States federal government. When we're talking about indigenous people globally, we're really talking about a fairly large fraction of the world's population. There's about 476 million people altogether. And so while commonly I'm coming at this from the viewpoint of kind of a North American-centric or even a US-centric stance, I think one of the first things we need to do is acknowledge the fact that there's a lot of places in the world where this is a much more pressing matter because the indigenous population in those countries is much more significant. Greenland's population is overwhelmingly Inuit, for example. So despite the fact that it's an entity controlled by Norway, Greenland is majority indigenous. And there's a number of other groups in other countries, Guatemala that we'll reference back to, which is almost 50% indigenous, Mexico being over 20% indigenous, with a number of different tribes still represented and still operating as independent entities there, all the way down to the United States, which only has a representation of about 2-ish% of its population. So within the United States, when we're talking about indigenous people, we're really talking about people that exist within the 574 currently federally recognized tribes that are found across 325 reservations and include around 5 million people. So that's about that 1 to 2%-ish. When we're talking about kind of this term of Native American or American Indian, though, indigenous groups in Hawaii or in Alaska, Alaska Natives and Native Hawaiians don't really get included in some of these same frameworks because the relationship with the US federal government is different. Alaska Natives are built around this corporation entity that was developed in the 40s and the 50s with the incorporation of Alaska. Native Hawaiians really don't have that federal relationship established in the same way that either Alaska Natives or Native Americans do. So I'm really going to be talking about the context of Native Americans within what we can think of as the lower 48. This idea relates to tribal nations, and this idea of tribal nations and their relationship with the federal government is codified under US law. There are statements within Supreme Court cases, such as the one that I've shown here with the Cherokee Nation versus the state of Georgia in 1831, that preceded removal of the Cherokee people from the southeastern United States into Oklahoma, demonstrates very clearly the way that the United States is recognizing different tribes, is this term of domestic dependent nations. And so the clear question is, okay, well, what does this mean? How does this define those relationships? And it really kind of centers around three ideas I'd like to put forward: governance, sovereignty, and treaty rights. Governance in the idea that tribes have the authority to establish and enforce their own laws. Sovereignty in the fact that they have the right to establish and define relationships with different entities. That could be the US federal government, it could be other tribal governments, and it can be other nations within the world. And that's happened before. And that these treaty rights outline the relationships that exist between the US federal government and those tribes, and they're codified by law, specifically around oftentimes services in land protection that provide those reservations that were mentioned on the last slide. That also means that they have the right to define different operations that exist within their boundaries, including things like the right to ban research. Now, I bring this up not as some theoretical exercise, but the fact that this has happened. It's happened quite a few times. And the one that I want to highlight here is specifically a moratorium on genetic research that's been imposed by the Navajo Nation for about 2 decades now, just over 2 decades. And this is important when we're thinking about the relationship between different tribal entities and researchers, because it establishes both a framework in which work can or cannot be done, but also influences more than just that tribal nation. And the Navajo Nation was specifically important within kind of these contexts because it's the second largest tribe within the United States, and it has the largest reservation landmass. So a lot of other tribal nations look to what Navajo Nation and some of the other large nations like Cherokee Nation of Oklahoma are doing to kind of set standards and guidelines for themselves as they move forward, because they understand that those larger tribes have resources that dictate their reasoning and their justifications for moving in different directions. And oftentimes those directions are going to be good guidelines for them to be following down the road as well. And you may be asking yourself, okay, well, why this ban on research within certain tribal entities? And I think it's important to say that this isn't necessarily the result of something new that's happened. This is really a buildup of repeated issues around scientific research with tribal communities, indigenous people worldwide. But to highlight some of the ways that this has happened specifically within North America and the United States specifically, there are some examples that I'd like to bring up here really quickly about why these bans on different types of research could be imposed. And it comes from lots of different ways in which science has directly negatively impacted different tribal nations. You may not have heard of any of these, but there have been a lot of studies that are similar in some ways to observational studies of infections around, for example, syphilis with Tuskegee and in South America, looking at how uranium exposure in different uranium mines on Navajo Nation affected Navajo people over time, right? So these were being conducted 1940s through World War II into the Cold War era. And instead of reducing harm to the people, that there was concerns about negative outcomes, they simply looked to see what the outcomes were. And so instead of some of these more passive observational studies, there have been some more direct and very targeted studies that have produced harm. The residential school system, for example, overall was quite harmful, but even within the residential school systems, there were specific experiments carried out by scientific researchers, sponsored by the state, in different areas. And some of the most infamous of these studies involved ESP investigations around the beginning of the 20th century. So individuals were being deprived of food and water and contact to try and push ESP kinetic abilities out of Native youth that were being held in these residential schools that were ultimately built with this model of save the man and destroy the Indian, right? So more general, more targeted, and if we're thinking, well, okay, fine, these have happened, but certainly these haven't happened anytime during our lifetimes or recent history. That's simply not the case. Were a number of studies in the 60s, 70s, and the 80s around this drug that's currently FDA approved for birth control called Depo-Provera. And it was tested on women in the 70s and 80s prior to that FDA approval to see what the outcomes and the effects were of this drug. And it was specifically tested around indigenous and recent migrant women, typically from South and Central America. So alongside those temporary sterilizations, there are absolutely full long-term sterilizations that occurred, but the Depo-Provera studies show specifically how the scientific community was involved in this. That misconduct stretches out into the present, and it stretches out into genetic research studies. And I probably don't need to spend too much time around this, as I'm sure most of you have probably encountered this at one point or another, but this case between the Havasupai tribe and the Arizona Board of Regents, specifically around misconduct with genetic material at ASU, is one of the most prominent and kind of clear examples of genetic misconduct between researchers and participating groups, specifically tribes. So Havasupai are a small tribe located at the bottom of the Grand Canyon, and because of that relative isolation compared to a number of other tribal groups that are more accessible, the Havasupai were considered kind of an ideal population for genetic studies. There's only about 600 tribal members, 650. And so as early as 1990, tribal members had reached out to Arizona State specifically because there were concerns around diabetes within the community, and they wanted to understand what they could do to overcome these negative health outcomes being experienced by their community. And as part of that, donated blood. Now, it became clear around 2002, 2003, a little bit by circumstance, by tribal members of Havasupai, that their research materials that were donated for diabetes research were not being used just for that, which they had definitely not consented to, and were being used for a number of different studies by researchers across the globe, no longer just at ASU, for things like migration, alcoholism, mental capacity, et cetera, studies. So this resulted in a settlement around 2010 where the Havasupai were provided the samples that they had donated to reclaim them, and a cash settlement and promises of additional resources provided by the state of Arizona. So this one case may seem like an outlier, but there's a lot of DNA projects that have targeted indigenous people over time. I need to bring up a number of these studies, including the Human Genome Diversity Project, which was specifically labeled as a vampire project, involving a number of different indigenous groups globally to try and get DNA from indigenous people, quote unquote, before they disappeared. This resulted in a lot of pushback and the production of a number of indigenous genetic councils. One of most notable is by the San people in South Africa, pushing back against the Human Genome Diversity Project. Another is the Genographic Project, which started in 2005, that basically had the same central tenet to its foundation, in response to the fact that the Human Genome Diversity Project wasn't able to move as far as it wanted to because of all this pushback that happened. These projects, again, are somewhat dated at this point. What I'd like to demonstrate clearly is that these projects are continuing in different forms. So there is a Helix Project, which is fundamentally a Genographic Project 2.0, and is actually referenced that way in a number of different publications you can find produced by the Helix Corporation. So again, the same idea, targeting indigenous groups globally to get DNA from them before, again, this idea of it would, quote unquote, disappear. And there's a number of other projects specifically within the United States that have targeted indigenous groups. One that's been very prominent in a lot of media is the All of Us project, sponsored by the NIH, which has the goal of collecting and sequencing the genomes of 1 million different citizens in the United States to get an idea of what the genetic diversity is within the population in order to apply it to personalized medicine. Another one that's been less talked about is the Million Veterans Project. And this may seem a little bit more innocuous because it's targeting former US personnel, military personnel. So as a former veteran myself, there is a clear trend within the military for overrepresentation of certain groups, and one of those being Native Americans and Alaska Natives. So indigenous people serving in the US military allows that overrepresentation and that access to different indigenous DNA that doesn't necessarily exist with all of us. And because the VA, the Veterans Administration, is the one processing samples through the MVP project, it's actually been very successful because it's just another form that you sign in order to receive your benefits. So the MVP project has already enrolled 825,000 people. Its initial target was 1 million. They've bumped that out to 2 million because of the massive success they've had in enrolling people so far, a number of which are going to be indigenous. So there may be a standard question or a standard pushback to this is the fact that, you know, at the end of the day, it's just some DNA. So what's the big deal? And I think it's important to highlight that it's a big deal in what I'm going to say is two strongly distinct ways, in addition to another number of ways. The first is in power. Having that material allows you to dictate the conversation. One of the major issues that the Havasupai had with the initial indication that their DNA was being used for other studies was in a thesis defense talk about human migration. The specific Havasupai member that spoke up first as part of that defense, where they attended and found out that their samples were being used for not the diabetes research they had expected, confronted the presenter and asked, did you receive authorization to use the samples for these purposes? The answer was no. Well, the tribal member went on, this contradicts our own stories around the Grand Canyon being the birthplace of the world, where a flood formed the Grand Canyon, people were seeded there, and as the waters receded, people came out of it. And that specifically contradicts, you know, this idea of Bering Strait migrations. So that power about whose voice gets heard and how those stories get told are influenced through things as simple as DNA. And I think that that story is a clear indication of how that can happen. The other is money. Here's a Global Markets Insight report that came from Monday, talking about how much money direct-to-consumer genetic testing markets are worth currently and are projected to be worth in about 7 years. And what you can see is that the current market value of around 1 billion, which is a couple of years old now, is projected out to reach 3.4 billion with a 12.8% compound annual growth rate. So this is a massive industry that generates a massive amount of money. And it's all based on something as simple as DNA sequence. Now, DNA can move. So DNA from those direct-to-consumer genetic testing companies can move to other markets that have clear monetary outcomes. GSK bought out in a $300 million agreement a number of different genetic results from 23andMe a couple years ago, and then Blackstone just finished signing a deal specifically with Ancestry.com to buy out the entire company for $4.7 billion in order to wrap all of those DNA informations into their pharmaceutical companies that they own. So that DNA has massive impacts within the financial world, and it is not simply just a source material. It has value in the power that it establishes, in the way that different stories are told, and in the way that any story can get told based on current financial systems globally. This has brought out a number of different voices raising concerns about the way that these DNA projects are working to collect materials. Voiced by a number of different people, many of these have targeted the All of Us research program, because it's directly tied to the federal government. And it's easier to criticize the federal government than it is private corporations, because federal governments are supposed to listen to the population that they serve, as opposed to corporations, which can brush you off as long as they're meeting dividends for their investors and able to sustain themselves. So oftentimes approaching the federal government becomes an easier way to overcome inconsistencies and wrongdoings that are seen through science. The federal government is not innocent within kind of this framework. This is an article that came out a few years ago in the Boston Review, talking about how the approach to treatment of different Native nations by the United States federal government makes it very easy for people that are interested in targeting certain vulnerable populations like tribal nations to further claim resources from them and hurt them in the process. So there's this imbalance of power that already exists within the federal government based on the way that the United States has typically dealt with tribal nations. And I would argue that what we've seen in the last week is just a continuation of those same ideas, right? It's a reclamation of ideas or value in places that people feel that they can take what they want in order to serve their own needs despite the voice of others. So again, these ideas of being able to take in order to gain access to what you want while hurting other people, especially people that are minoritized or are underrepresented, is kind of a contiguous story of the United States, both in practice and as we're seeing oftentimes in the mentality and the principle that's kind of dictated by the population itself. So what I'm highlighting here with the United States is not restricted only to the United States. Again, it's important to point out that this is not a specific United States problem. This is a global problem. We see this through a number of articles, some of which I'm just highlighting here. I mentioned specifically issues around South America. In Bolivia, following the departure of Evo Morales, who was the first head of nation indigenous person in the Western Hemisphere, his departure and his fleeing to Mexico brought a lot of pressure back on indigenous groups within Bolivia, where they're being persecuted both in their rights and individually with disappearances and targeted assassinations in order to quell kind of that voice that had risen up. Beyond the idea of controlling a people has been direct assaults on property. This has occurred very recently in Canada around Micmac lobster fishermen and Canadian fishermen, where Canadian fishermen set on fire two of the Micmac warehouses of their lobster catches, moving from this idea of hurting a group of people to very specifically targeting their economic and their viability. And then this does come down to the individual level with the disappearance of different members. So a Karen activist whose village was encompassed in a new national park that was developed by Thailand was disappeared by the head of the Parks Department in Thailand just about a year and a half ago. And his body was discovered a couple months later. So I've been bringing up a lot about people, and I know that the topic of this talk was really about microbiome. And it's clear that there's a lot of issues revolving around performing human research. And so one of the easy ways to oftentimes not have to deal with some of these intricacies is, instead of working on a human, to be able to work on human-associated contexts, things like the microbiome, right? And what I'm going to try and demonstrate to you is that the work on human microbiome research really parallels the work on human research. And then I'll talk about some of the specific ways that we're approaching microbiome research now to be able to address these disconnected relationships between researchers and tribal nations. So the microbiome encompasses a lot of different organisms that are kind of showed here in this cartoon. And the microbiota, those organisms that compose the microbiome, are really all of the cells living with us that are not specifically us. So with human that are not human, or with sheep that are not sheep, or with pig that are not pig. The microbiome is really important because over the last 10 years, it's become very clear that these cells are diverse and make up a large kind of organ system with the human body. The organisms talk to us and they talk to each other, dictating health and disease, and they can change with our environment, right? So these genes can tell us a lot about what's going on around us, and it can tell a lot about what's going on with us. Some of the best indicators of human health for different disease outcomes have actually been the microbiome, and I'll speak directly to that later. The microbiome is also a really nice, tangible target for scientific research because of how vast it is. Compared to humans, the microbiome is estimated between either an equal number of cells as exists within the human body or in excess of 10 times the number of human cells. And now it's starting to get walked back to probably being closer to an equivalent number of cells as the human body. But what has stayed the same through that walk back in the number of cells is the number of genes that exist within the microbiota of the human system. There's approximately 100 times more genes within the microbiome than there is within the human. So if we think about how much work is being done within human genetics or genomics, the ability to work on concepts around cell biology, microbial biology, physiology, genetic communication, genome and evolution within the microbiome is vast. It's orders of magnitude larger. So studying the microbiome research allows you to raise fewer concerns around misuse because it's not human subject research directly. You get the ability to collect samples and analyze data with pipelines that have been installed over about the last 15 years that really helped kind of bring the field to maturity to the point that now you can run cross comparisons against studies. With Indigenous people specifically, there's often unique environments in which Indigenous groups reside that are different from their industrialized counterparts globally and specifically within a single nation. A number of papers have demonstrated how those Indigenous people's microbiomes, being different from a number of industrialized people, provide kind of this resource that's been underexplored relative to most human microbiome research that could have potential in human health and disease. The microbiome field is still quite new, again, only being about 15, 20 years old, so there's a lot of room for growth here. What I'd like to highlight before I move into how to conduct or how I would suggest or approach this microbiome research is the fact that microbiome research is still human subjects research. And nothing makes this more clear than looking at some actual microbiome data that we have from a COVID study that we're running with Columbia University right now on patients from NYU's hospital system. So these are lung exudates taken from sputum in COVID patients that were intubated as part of the initial wave of COVID-19 through the United States. And this is total RNA that we've collected from these patients and then mapped back to both a human genome reference, HG38, as well as a database, a Kraken database, specifically of roughly 36, I think it's 4,000 bacterial species and approximately 1,000 eukaryotic microbes. And what you're seeing is that the overwhelming majority of the sample that was recovered from these patients is human, right? So when we're doing microbiome research, even though the interest is focused on the bacterial cell or the eukaryotic microbe cell, the potential to recover human material from these studies is massive. While this isn't always the case, this is kind of one of the extremes. This being another case example where now we're looking at stool samples collected from some Moroccan participants with some colleagues there in Morocco, where there's a lot less human material coming through. We're still sequencing roughly between 40 and 200 million reads for each of these samples, which means at the end of the day, a 5 to 10% human contamination gives us about 20 million reads for a human sample, which is still a lot. So microbiome research, because of the fact that you recover host material every time you do this, is still human subjects research. So the way that we approach microbiome research needs to be compatible with the way that we would do human subject research. The expectation as a researcher would oftentimes be that working with tribal participants and tribal nations would be analogous to working with any patient population within the United States. You as a researcher would go out, meet with an IRB, that IRB would then authorize you to begin the study, and you go out and you recruit tribal participants. The reality of the situation is something that looks quite a bit different. Anybody that's tried to do this is going to find out that you're going to go through multiple IRBs. Typically, you're going to go through your university IRB, you're going to be going through your tribal IRB, and you could be going through an Indian Health Services and IHS IRB as well. So you're going to be cross-talking a lot of different oversight bodies in the process. Meanwhile, your university is going to be talking with other federal entities to ensure that the research process is being done correctly, such as IHS. IHS is also constantly cross-talking with tribal members and tribal officials. And then at some point, the tribal participants are recruited, but you're not really working to the tribal participants typically through one link. It's through multi-links. So your ability to kind of oversee the study the way you normally would with a non-Indigenous population within the United States looks very different. And this has kind of led to the impetus or push for broad consent when working with different tribal participants that isn't always quite as prevalent when working with different populations in the United States. Because of pushback on broad consent forms by tribal nations as a result, there's a lot of impetus for looking at ways to circumvent sovereignty, disillusionment by researchers, or kind of animosity, this us versus them that gets set up between research and different tribal nations. And so there's a lot of hindrances that exist for different researchers to engage Native communities. There's a lack of training and understanding what these structures are going to look like when you start to approach them. It requires a lot of time because you have to navigate all these complex relationships to build a really good working relationship with the tribal entity to continue those studies out. Some tribes have a relatively small N. As I mentioned, the Havasupai was only around a 650 person community, which presented really nice for some of the genetics, but also means that you had have to establish a really good working relationship to get enough participants to be able to run many of the studies you would need. And some of the descriptions of unsuccessful attempts, such as the one I already mentioned, can discourage researchers from reaching out to tribal participants at all, because there's this feeling of, well, what's the point? Because I'm going to get shot down anyway. What I'd like to demonstrate, there's frustrations in both directions. Indigenous participants are also going to be feeling frustrated with this process because oftentimes there's this feeling of participation without benefit. Where is any of this going to actually affect either the individual or the community or other tribal communities that exist within the United States or globally? And a lot of this is based on historical context. Attempts by different entities established by the US government or not to define political status of Indian based on DNA and set up enrollment and blood quantum designations that either remove tribal members that are integral to the community or add different segments of the community that maybe weren't integrated before, this idea of diluting out some of the benefits that exist within a tribe. There are concerns of stigma, specifically around some of the research that I mentioned with the Havasupai, the inability to truly de-identify individuals because of the small ends that exist within some of these communities. The ability to negate traditional knowledge systems. The story I mentioned about the Havasupai, specifically around human migration, again says, well, your story is wrong, our story is correct. And that overrides traditional systems that exist within the community. The inability to return the results, so oftentimes the materials go out, but no information comes back. And all of these have kind of set up a dichotomy between researchers and tribal participants that revolve around who owns the data and who gets the benefit. So these concepts of biocolonialism, you're coming in, you're scavenging what you can from us, and we see no benefit from them. And so data systems have reinforced this, which is incredibly problematic, especially around microbiome research. Typically, what you're dealing with is when a participant provides you with that information, you contracted by government funds through the NIH or NSF, or through private entities that also kind of have these established relationships about what happens to the data when it's collected by the researcher, means it's going to get deposited into a federal data bank. It's going to end up in GEO or SRA or something like that, with the established purpose of being able to distribute it back out to researchers so that there's more good that can be produced from a single data set, which makes a lot of sense. The problem with this is it sets up a gradient at which, as soon as that material leaves tribal participants, there's no ownership left. The issue that revolves around this is, again, reaching back to that governance and sovereignty concept. tribal participants are members of a tribal government. They're members of a domestic dependent nation. Their relationship with the US federal government is dictated very specifically. They're not just a segment of the US population. They're specifically designated as different within the US population. And so these data systems and the way that we're pushed to open access data actually hinders tribal participation because of that open data access format. There are also issues around individual consent. The ability to deposit that data is because individual preference and the ability for that individual to say, yes, go ahead and use my information, is really what gets overriding precedent within the United States. And this is fine, but it becomes very problematic within a small population, where the ability to pick out an individual within a larger group become easier and easier. I would argue as a result, when working with Indigenous groups, we need to be thinking instead of individual consent, but as a multi-layered consent framework. And the WHO has actually set up this framework already, which is great. It requires individual consent, community consent, so this is often going to be occurring at the tribal level, and a wider Indigenous organization consent. And the reason for this is because of that issues around ownership, the issues around governance of the data, the issues around benefit of the data don't affect just the individual, it affects the community and it affects Indigenous people more broadly. When we're talking about wider Indigenous organization consent, some of that can look like for the United States, the National Congress of American Indians, which has been specifically involved with all of us and is the one that really stopped them in their tracks on collecting Native American samples within the United States because they withdrew and prohibited their consent to the gathering of any more Native samples. This idea of consent really gets at a statement from Kim Tallbear where she highlights why we're looking for some of these consents. And I've already talked about some of these ideas here, but she states it really succinctly. So these concepts of who gets to tell the story, anthropologist, geneticist, or the community. And that ability to tell the story informs law and policy. This informs the way that that community is going to be able to operate down the road. And the voice gets disseminated more broadly to the US general population, the global population, that sets up the way that that population is viewed by everyone else. So to kind of help set up a better framework for working with Indigenous communities, a couple of years ago, we published this paper that highlights 6 guiding principles. So they're all listed here. I can let you read them out, but they all center around this idea of sovereignty, maintaining that sovereignty with tribal nations, and really engaging the community on their own terms, which sets kind of the title for this talk. In order to carry out these 6 guiding principles, there's kind of 2 models for research right now. One is working in tribal trust relationships. So this is standard academic or federal practiced research. Tribal participants aren't actually involved in this research too much. It's somewhat extractive. There's no ownership of the data by tribal participants. They may get some of the results back, but there's no ownership, no governance by tribal members. And the consequences of misconduct within tribal nations are usually really minor in this context. So there's going to be a pushback to this by different tribal people. The other is tribally driven research, but there's a lot of issues in tribal people trying to put forth research projects right now based on the way that, at least in the United States and Canada, research contexts have been established, where you have to show that you have the resources in order to be able to perform the research, right? You can't just say, I'm going to do the study without having the established framework, that equipment and facilities in place to be able to do it. Also, specifically, government interest can change over time. So one tribal government can be interested in running a study, but the next tribal government that comes in can completely squash the study, whereas academic and federal institutions are buffered from being able to have changes in administrations change research that's ongoing. And so what we're kind of proposing is a third model. And this is tribal partnership. What I mean by that is an independent entity that is tribally owned, staffed by tribal members, but operates outside the framework of the tribal nation. So exists as an outside entity, a corporation or a nonprofit that can attract funds specifically, but yet provide that oversight by tribal members, by tribal council, in order to make sure that that issue of ownership is retained within the tribal nation. So the results are open to the tribe. They are not dictated by tribal government turnover. Instead, they're dictated by funding contracts through granting with federal governments or private corporations and institutes. I want to say that we've established one of those recently. The Native Biodata Consortium is an independent 501c3 that's BSL2 certified and can perform fundamentally any molecular assay you throw at it. It has informatics infrastructure associated with it as well. So it has all the components of a research lab. It's housed on the Cheyenne River Reservation in South Dakota in the KEA building. And it's advised by Indigenous people. Its entire executive board is Indigenous. It retains additional local legal advisors that are Lakota themselves. And it's guided by a community advisory group of Cheyenne River residents. So this gives us the full framework in order to kind of check off the boxes of being able to do good molecular work, give data sovereignty back to tribal nations, and engage community in these talks the entire way through, because it incorporates tribal members at all levels. There are other efforts in place to be able to train people to staff these kind of positions, and I want to highlight two right here. The Summer Internship for Indigenous People in Genomics, SING, that's been going for about a decade now, and has trained about 120 different Indigenous participants from around 60 nations within the United States. It's expanded globally at this point to Aotearoa, or New Zealand, Australia, and Canada, with current plans in the works for establishing a SING Chile in the next year or two. It's produced high-impact guidelines that have appeared in Science and Nature Communications, and it has a trainee investigator support system for Indigenous people as they're moving through genetics PhD programs, postdocs, faculty positions, and industry positions. The other is Indigidata, which we just initiated this year, that's really going to be focusing on informatic training in biological systems. So large data set analysis, as well as microbial community and other ecological data set analysis. And this is being performed on site at NBDC. So once we have these tribally driven research entities that are independent, things like NBDC, we can really expand out the toolkit in which we can build out more of these systems for tribally dictated research programs. One I want to highlight here is called SAIL, or the Studies of Autoimmunity in the Lakota. This is a project to study why high levels of rheumatoid arthritis and autoimmune disorder are prevalent in Lakota people. This is a project that was instigated within Mike Snyder's lab at Stanford that we've been able to contribute to, specifically to look at the microbiome components of Lakota people in its association with RA. So if we look at the United States, there's a 2 times higher incidence of rheumatoid arthritis in South Dakota. So this suggests that there's got to be some kind of environmental pressure or agent there predisposing people or pushing people towards an RA manifestation or phenotype. Within South Dakota, on Cheyenne River specifically, there's actually a 5 times higher incidence of rheumatoid arthritis, which again pushes at this idea that there's got to be either other environmental impacts in which Cheyenne River is being acutely affected, or there could be genetic components. So to get at this, the Snyder Lab is really investigating the entire swath of what you can do in a multi-omics approach, looking at genomics of participants, proteomics, transcriptomics, metabolomics, environmental sourcing, and our role within this, the microbiota, or the microbiome of people on Cheyenne River that have rheumatoid arthritis versus those that have not presented with that clinical manifestation. The reason that I think the microbiome is going to be particularly important in this study is because if you look at the progression of RA, not necessarily just within Cheyenne River or Indigenous people specifically, but more largely based in RA diagnosis. Prior to RA being diagnosed clinically, there's a change about 6 months prior in the microbiota of those individuals that kind of gives a hint at some autoimmune disorder arising, suggesting that the microbiome is intimately tied to diseases such as RA manifesting. And this becomes even more acutely important because there's higher incidence of RA within Cheyenne River. Microbiome, as I mentioned before, varies based on lifestyle and context in which a person finds themselves. And this study that was published a couple years ago in Science really highlighted that very well. So what we're seeing here is we're seeing a scatter plot of every microbiome study that's been done up to that point and where they exist on a PCOA plot. You can see some major taxonomical changes on the X and the Y axis as you move left to right or up and down. The major axes, PCOA1 here, is based on how industrialized you are, where more industrialized groups tend to cluster more to the right on the graph, and less industrialized groups in traditional societies cluster more to the left. So it suggests very clearly that there's microbiome variation based on lifestyle. And if we're thinking about this from a non-bacterial-centric viewpoint, we can see that even more clearly in a couple different ways, where indigenous South Americans that have been involved in microbiome studies have high levels of microbial eukaryotes within the GI, whereas the US population, protozoans, helminths, the answer is usually there are none. Occasionally there will be a protist, specifically Blastocystis, and then there's often some fungal stuff, either Saccharomyces or Candida. Now within RA, we have the hypothesis that eukaryote diversity is actually going to be really important here, specifically because of the immune response that's generated during RA. It's a Th17 response that is specifically often in place around mucosal barriers to restrict eukaryotic overgrowth, oftentimes fungi, but not just fungi. When fungi are sent to overgrow those mucosal niches, they're sensed by the immune system, which activates a Th17 response. That Th17 response can migrate to different parts of the body. Specifically, it often can migrate through to joints. Now, specifically around joints, rheumatoid arthritis is an hyperinflammatory response mediated by Th17 at those joints, producing this kind of feedback of inflammation that occurs there. So as we're moving forward on the microbiome research, we're being very mindful of the microbial eukaryotes that exist within different people because of this tie to Th17 and how that can lead to rheumatoid arthritis. Within microbiome research, most eukaryotic studies have focused on amplicon-based sequencing, specifically ITS. There's a problem with ITS. It's part of the ribosomal array, as I'm depicting here, but only recognizes about 40, the primers that are usually used in these studies only recognize about 44% of the known species that exist within the GI, which means we're missing a lot. There's also a massive amount of database variability. There's no standardization for eukaryotic species as there has been for bacterial species. And there are different PCR biases based on read lengths of the ITS regions and where those primers bind. And so what I'm showing you down here at the bottom of the screen is identical individuals, identical samples that are paired. So each set of 2 bars is the same sample. It was assayed for eukaryotic species, either by using amplicon sequencing of ITS1 or using amplicon-based sequencing of ITS2, which is the other kind of quote-unquote gold standard for measuring eukaryotes right now. And hopefully what you can see very clearly is that there's a lot of discordance here, which means there has to be a better way to do this, because right now, really we're shooting in the dark when it comes to microbial eukaryotes. And so we've been working on developing a metagenomics approach. We use a defined microbial community, 5 bacterial species, 5 eukaryotic species, and then we start them off at a defined proportion. I bring up the pointer here. We see them where it's 99% bacterial species presented by these warm colors and 1% eukaryotic species, which is what our microbiomes, the standard industrialized microbiome, is thought to look like. And then use our protocol to see if we can enrich for eukaryotic species. And what I'm showing here is 2 steps in our processing in which we're able to pull out the eukaryotes within this overwhelmingly, again, warm color bacterial centric community and dominate the community by eukaryotes, these cool colors here, by the end of our protocol, which means we can begin getting at the eukaryotic species. Some of the other issues with running a metagenomics approach with microbial eukaryotes is the fact that databases currently don't exist to be able to handle these kind of data types. Because if we're looking at the standard Kraken database for running metagenomics on bacterial species, it has 32,000 bacterial species, has a fair number of archaea, but the fungi, protozoans, and helminths are really poorly represented. And there's no other approach outside of running Kraken right now to be able to even handle metagenomic sequences for eukaryotic species, because each genome is too large, and the reference library size becomes overwhelmingly large. What we've been able to do is we've been able to build an in-house database for microbial eukaryotes by scraping JGI, the Joint Genomes Institute, as well as NCBI's database, and build a much larger database than what Kraken shows here, especially around fungal members. This in-house protocol, using a approach called SMART, which is the only one that can handle a library size of roughly 2,000, 2,500 eukaryotic genomes, allows us to begin mapping metagenomic sequences back to eukaryotes. And so if we test that on metagenomic sequencing of some different stool samples we've collected, the expectation is, again, 99% of that stool is probably going to be bacterial, 1% is going to be fungal eukaryotic. We can begin pulling out many, many more eukaryotes and archaea from these samples than we could previously. We're still working on some of the optimization, but we know that we're pulling out microbial species accurately because we can take these metagenomic profiles and then align them to specific reference genomes. So we've done that for some species, including Candida albicans, which is a standard intestinal resident, and we get good concordance between mapping with our protocols and by doing target mapping just to the Candida genome reference. This has allowed us to identify a lot of new taxa within the GI. These are all different microbial eukaryotes that we've identified within humans, specifically industrialized humans, where you wouldn't expect this sort of diversity, which is great. It means that we can begin moving into the study specifically around RA with indigenous folks and be able to look at both the diversity in the bacterial species as well as the eukaryotic diversity moving forward that we think may be at the root of some of these autoimmune diseases. So I want to leave you, as I'm trying to wrap up pretty quickly, with, well, what comes next? And I think we need to highlight some different things here. We need to support indigenous people at all levels and steps of scientific inquiry. This includes students, it includes tribes and tribal members as we build out research plans. So we as researchers really need to be thinking about how we decide what science we're going to be doing and how we're going to be approaching the work and its dissemination, because we're oftentimes excluding indigenous people in the process. Specifically around the science, experimental and computational aspects of microbiome work still exist that are left unresolved, that need to be addressed. And these need to be resolved because hopefully what I've been able to show you in a couple slides is we have a large segment of the microbial community that's not being involved in the research or the documentation of potential associations with human health. So we're not getting a complete view of this complex community. So that, I just want to acknowledge a number of folks, the MBDC board that's kind of listed here, specifically Crystal Sosi for inviting me to this talk that you all have been present for, and thank you for your attention. The same program and a number of the coordinators there and the faculty that I've listed here. My lab group, including specifically Otter Crouch, that's been involved in a lot of our microbiome work with Cheyenne River, and Molly Rambeau, who's a Cheyenne River tribal member who's here at Ohio State and has been involved in the project since she was a sophomore. So finding ways to incorporate tribal members specifically in your research is very possible, as well as our collaborators at Stanford that really kicked this project off in the first place. With that, be happy to take any questions.
All right, thank you very much. That was fascinating. Let's see if we have any questions. You can type them in the chat. I think you can unmute and ask now as well. I recognize we only have a couple of minutes before the top of the hour, so let me ask while people are figuring out how to ask themselves, if you can give us a little bit of insight into the capacity building work you talked about. So with respect to, let's say, graduate education or other things that would be helpful. Can you tell us a little bit about efforts or needs in capacity building for Indigenous data science research?
I think training is an obvious one, which is why this Indigidata seemed like a clear outgrowth from some of the genomics work that's been happening for about 10 years. There's simply not a lot of Indigenous data scientists. And I think a big part of this is because it's not clear to a number of students as they're coming through the system how that would be applicable to their home communities. Even though now that you've been in science long enough, it becomes fairly obvious, okay, well, survey information, ecological information is all data, and that has clear impacts on community. The other piece that I think we need to be thinking about is data storage and management. So if tribes are going to be able to hold on to their own data, what is that infrastructure going to look like? Because there's clearly value tied to that data. So what safeguards can data scientists help put in place for tribal partners so that they can safeguard the information they're trying?
I'm a little surprised you didn't talk about money. Where does the resources come from to do this capacity building?
That's a good question. So the efforts in training that we have are all based around federal granting. So we have an NIH grant that specifically funds SING, and we have an NSF. It's attached to Career Award that's funding the data science portion. We've been very lucky and able to get some corporate sponsorship as well. So Amgen has actually helped sponsor most of this year's workshop for the data science bootcamp. So there are partners and there are pots of money out there. And I think there is an interest in corporate, nonprofit, and federal entities in supporting this work. It's just making it clear that you have, again, that framework that they're all going to be asking for to deliver on what you're asking to do, and not just saying this needs to be done without context.
Great, that's very helpful. There's a question in the chat from Ping Zhao Hu at the University of Manitoba, and the question is, to publish or present any Indigenous-related research, we are required to get approval from tribal-related agencies first. Is this true?
That is the correct way to do this. So a good working relationship would have tribal oversight get an idea of what is going to go out into the world before you put it out either into an archive or you present it for publication. There are certainly instances where that is not happening. But again, if the goal here is to move towards a more ethical balance in the way that we're approaching our research, that should absolutely be part of the process. It's not mandated by law by any means.
All right, well, we're a little over time, so let me, Matt, thank you once again for this really interesting and helpful presentation. It's been recorded. Oh, there's one more question, so if people will stick around, let's do it. This is from Wasif Khan, and his question is, thanks for the talk, Dr. Anderson. You mentioned that ITS1 is accurate for about 44% at species level. Is the classification better at the genus or family level?
Yeah, that's a good question. Probably. So that 44% number is not based on reference to metagenomic sampling. It's actually reference to visual sampling, which is how microbiome work was done before next generation sequencing was possible. So the identification of microbial residence through visualization, which happened prior, may not have differentiated species. So it may have been at more of a taxa or family level, but the resolution is probably going to be a little bit better if you move farther up.
All right, and there's another one. Your questions have been very active. Ricardo Mendez from the University of Puerto Rico School of Medicine says, how are tribal member scientists represented in the genomics research community, if at all?
Sure. So it's a small community where most people know everybody else or close to it. So the representation is really low. Within genetics, I can place where I am in the single digits for those having acquired a PhD. So within the United States, I was the 5th Indigenous person to get a PhD in genetics, and I know everybody that came before me except for one personally, and I know personally everybody that's come since, because we're just peeking into double-digit numbers now. It's small. How have they organized? So a lot of us are starting to organize around some of these places that we're trying to push better engagement. So SING has been an outgrowth of kind of that attempt to organize us. Indigidata and this Native Biodata Consortium is another attempt to kind of organize ourselves around kind of our focal topics, our scientific topics of interest. As somebody that also practices microbiology for the most part, there's also a lot of interest around the few microbiologists that are Indigenous to be able to organize ourselves. And these networks are slowly expanding globally. So SING has been really instrumental in this, in connecting folks here within the United States to genomicists and geneticists in Canada, to geneticists and genomicists in Australia and Aotearoa, New Zealand. So these networks are expanding, but it's slow and it's taking time. And a lot of that is because the representation is so low.